everyone's Gaussian this side. So today hand we are dealing with the final portion of the cellular respiration, that is called as electron transport chain or electron transport system. So let's get started. But beforehand, if you are literally going through all of them, you have to deal with the glycolysis and the Krebs cycle as well. Okay? Deal with. Now, all the NADH plus H plus molecules or the FADH2 molecules which have been synthesized in the glycolysis, in the link reaction and in the Krebs cycle will be cached here in the electron transport system. So basically the electron transport system and the complexes in the ETC are founded on the inner mitochondrial membrane. So here what I have gone for, this is the matrix of the mitochondria, this is the inner mitochondrial membrane and this is the intermembrane space, which means after this intermembrane space we will be having the outer mitochondrial membrane. And there are 5 complexes which are combinedly preparing ATP molecules. Okay, deal with. Now, dealing with the names of the complexes. So here the complex number 1. The complex number 1 is called as NADH dehydrogenase. The complex number 1 is called as NADH dehydrogenase. Okay, the complex number 1 is called as NADH dehydrogenase. Deal with. The second number complex is here complex 2. The complex 2 is also called as FADH dehydrogenase or succinate dehydrogenase. Okay, deal with. Then dealing with the complex number 3 here. And the name of the complex number 3 is none other than cytochrome BC1. Why? Because we have cytochrome B as well and the cytochrome C1 as well here in the complex number 3. And the fourth one is called as a cytochrome C oxidase. Complex number 4 is called as cytochrome C oxidase. Okay, deal with. And the last but not the least one, the fifth complex is called as ATP synthase. Some of the books are not accepting the fifth one as the complex, but still we will be calling the fifth one as the complex number 5. So deal with. Now, the first one is the NADH dehydrogenase, which means the reaction here will be taking as NADH plus H plus, which has been formed by the Krebs cycle in the mitochondrial matrix, is coming to the complex number 1 for making the ATPs. So here the oxidation of the NADH plus H plus will take place, or rather I will be calling it as dehydrogenation of the NADH plus H plus will take place, and the NADH plus H plus will be converted into NAD. And the remaining H plus ions are directly transported out of the matrix and in the intermembrane space through the complex number 1 protein. Okay, and due to the result of the hydrogen ions or the protons, some electrons are also evolved here. And these electrons will be following the pathway of, first they will be entering to the FMN unit, then they will be dealing with the FES, ferrous sulfate, flavin mononucleotide, then ferrous sulfate, and then these electrons will be going to the ubiquinone. That is what we call as UQ, ubiquinone. Then they will be entering into the reduced form of the ubiquinone, that is called as ubiquinol. Please do remember one thing, UQ is ubiquinone and UQH is ubiquinol. Okay. Then after this ubiquinol, these electrons will be again going to the cytochrome. They will be again going to the cytochrome B, and from cytochrome B to FES, ferrous sulfate, then cytochrome C1. That is the reason why the complex number 3 is placed in between the complex 1 and the complex 2, because it is the common pathway for the electrons flowing from the complex 1 and the complex 2 as well. Maybe like you will be asking the question that why the complex 3 is in between of the complex 1 and complex 2 and not after the complex 2, because the complex 3 is the common pathway for the electrons from the complex 1 as well and from the complex number 2 as well. You can observe this from here. Like whenever the FADH2 is getting converted into FAD, here also we are having the electrons evolving. They are flowing the pathway from the FAD to ferrous sulfate, then ubiquinone to ubiquinol, and again they are reaching to the cytochrome B. And from the cytochrome B they are going to the ferrous sulfate and ultimately from the to the cytochrome C1. And after reaching to the cytochrome C1, this cytochrome C will be accepting this. Basically, this cytochrome C is acting as a shuttle which is running from this to this to this reaction. Okay, in this fashion. So this cytochrome C will be taking the electron from the complex number 3 and it will be directly giving the electron to the complex number 4 here. And from here, these electrons will be dealing with the copper A. Here two electron acceptors are present. The first electron acceptor will be called as copper A and the final electron acceptor will be called as copper B. But the pathway will be, they will be flowing from the copper A to cytochrome A, then cytochrome A to cytochrome B, from the cytochrome B to the copper B. And the energy released from these electrons will be responsible for the conversion of 1/2 oxygen and 2 hydrogen molecules into one water molecule. So the released energy from the electrons is being used to form one water molecule. Okay, deal with. This was all about the electron transport. Rather, we will be dealing with the oxidative phosphorylation as well. So here, like the 4 H plus ions have been generated from the dehydrogenation of the NADH plus H plus, and these will be coming here as well, and they will be directly going to the complex number 5. And if you will be observing about the complex number 2, here the complex number 2 is. If you will be observing about the complex number 2, here the complex number 2 is. So ultimately you will be dealing with that the complex number 2 is not a transmembrane protein, rather it is a peripheral protein. Rest all of them are integral protein, but the complex number 2 is the peripheral protein, and hence the complex number 2 has to give up its proton as well to the complex number 3. Here I will be drawing with the another pen. The protons are also following the same pathway, but still they will be following in this fashion. Correct? Okay. So here again one extra H plus ions you are getting, and these 4 H plus ions they are like much more here in the intermembrane space, and they want to go into the matrix, but they are not able to find any place to go there. So ultimately they'll be knocking on each and every door, and they'll be finding a proper door here. This is our ATP synthase, which is the only one which is accepting the H plus ions or the protons to be pumped in the matrix. Okay. At the same time, the complex number 4 is also pumping 2 protons. At the same time, the complex number 4 is also pumping 2 protons. You can observe. Okay, deal with. Now, if I will be dealing with the case, which means a lot of different protons have been generated here in the intermembrane space. So basically here the electrochemical gradient in the intermembrane space will become positive. And comparatively the electrochemical gradient in the inner membrane or the matrix will become negative. So basically, like the law, according to the law of concentration, everything flows from its higher concentration to the lower concentration. So these H plus ions will be trying to go out or go in the matrix. So ultimately they will be seeking help with a special complex called as a complex number 5, or what we call as ATP synthase. And what happens in the ATP synthase? It is basically made up of 2 different subunits. The first one is called as a F0 and the second is called as a F1. The F1 is towards the matrix, whereas the F0 is towards the intermembrane space, and both of them are situated on the inner mitochondrial membrane. So this is a path of the hydrogen, this is a path of the proton which will be following something like this. And these are ATP synthase is basically a spanning one. So ultimately whenever the protons are going through it, it will be spanning like a dynamo in the electric generation, and it will be producing a special kind of energy which is responsible for the formation of 1 ATP molecules from ADP and orthophosphate. Done? Okay. Now the question arises is how many hydrogen ions or how many protons are required to form 1 ATP? Which means how many protons are required which are spanning the ATP synthase F1 unit to form one single ATP. So the answer is here 4 protons are required to form 1 ATP. 4 protons are required to form 1 ATP. So please deal with the cases. Hopefully this lecture has been beneficial for you guys. Again repeating the names of the complexes. The complex number 1 is called as NADH dehydrogenase. The complex number 2 is called as a succinate dehydrogenase or FADH dehydrogenase. The complex number 3 is called as cytochrome BC1 because it has cytochrome B as well, it has cytochrome C1 as well. And the complex number 4 is called as cytochrome C oxidase because here the oxidation is taking place. That is the reason why we are calling it as cytochrome C oxidase. And the path of the electron here will be from the ubiquinone to ubiquinol, from the ubiquinone to ubiquinol, ubiquinol to cytochrome B to cytochrome C1, from cytochrome C1 to cytochrome C, and from cytochrome C to copper A, cytochrome B, and the copper B. And ultimately it will be forming a water molecule, whereas the protons are going from complex number 1 as well, complex number 2, 3, 4 as well. And they are only entering in the matrix through the complex number 5, and whenever they are entering, they are spanning the F1 ATP synthase subunit, and they are producing a special type of energy which is responsible for the conversion of ADP and orthophosphate into a high energy molecule called as ATP. And the complete formation of one single ATP, it needs 4 protons. One extra thing, one NADH molecule forms almost 3 ATPs, and one FADH2 molecule forms almost 2 ATPs on an average. Okay? Now, hopefully this lecture has been beneficial for you guys. Thank you very much for your so much support. If you are having any kind of doubt, you can ask me there in the comment section. Thank you.